Hello everyone, welcome to MMBS111 Introductory Microbiology. This is mini lecture 13.3. We're going to be discussing stage 2, the presentation of antigens. There are 3 learning outcomes for this mini lecture. First, list the characteristics of antigens that optimize the immune response. Second, define the following terms: hapten, alloantigens, superantigens, and allergens. And finally, describe the actions of antigen processing and presentation and the cells involved. All right, so once our B and T cells have differentiated, then we're going to present these cells with antigens. Remember, we've already discussed immunogen and antigen are synonyms. They mean the same thing. These are substances that can elicit or stimulate an immune response, and they have to meet certain criteria as far as foreignness, shape, size, and accessibility. And they must not be a normal constituent or component of the body. Some examples include whole microorganisms, parts of microorganisms, cells or substances that come from other humans, animals, plants, and various molecules. Most antigens are going to fall into the following 6 categories. They are proteins and polypeptides, so things like enzymes, cell surface structures, exotoxins. They may also be lipoproteins, like the components of cell membranes, glycoproteins, like blood cell markers. They may also be nucleoproteins, DNA that is combined with other proteins. They may be polysaccharide, like the components of bacterial capsules, and they may also be lipopolysaccharides, especially those components of gram-negative cell walls. In order to initiate a response in the immune system, the immunogen has to get the attention of those cells that monitor the body for foreign substances. So in order to initiate a response, those substances need to be large. They should also be complex macromolecules. Simple molecules that are repeating structures, so a repeating polypeptide chain or a repeating saccharide, they're not going to get the attention of the immune cells like large complex macromolecules do. Size is not the limiting factor. Again, they need to be complex. So these should be compounds that include a number of different biomolecules and not just repeating sequences of the same biomolecule. We've already discussed epitope in previous sections. The epitope is the part of the antigenic molecule that is recognized by the lymphocyte, and it's the primary signal to the immune system that this object or structure is foreign. A hapten is a foreign molecule that is too small to stimulate the immune system by itself. So these molecules are typically linked to other carrier molecules to increase their immunogenicity and their ability to develop or initiate an immune response. There are several other types of antigens mentioned in section 13.3. Alloantigens are surface markers and molecules that occur in some members of the same species, but not in others. Really good examples are the marker molecules that determine an individual's blood group. Also those major histocompatibility profile markers on self cells. And these alloantigens are responsible for tissue donor rejection, reactions that occur during blood transfusions, because the immune cells are recognizing those cell surface markers that are present on the new tissue or the blood cells that are not possessed by self. There are also substances known as superantigens. Typically, these are toxins that are produced by bacteria. They can stimulate a hyperactive response from T cells. And when that happens, the T cells are going to produce an excessive amount of cytokines, and that will lead to cell death and tissue damage. There are a number of autoimmune diseases as well as toxic shock syndrome that are associated with superantigens. Then we also have allergens, and these are antigens that will stimulate an allergic response. We're actually not going to discuss allergens in this course. If you want to learn more about allergens, you can read chapter 14. It's all about allergens and allergic reactions. All right, so antigens in most situations must be presented to T cells in order for a sufficient immune response. So there are several different types of antigen-presenting cells I've already mentioned. Our macrophages, B cells, and dendritic cells are all going to present antigens to T cells. So these antigen-presenting cells are going to process the antigen internally, and then it will be bound to that MHC2 receptor that is then going to present that antigen. It's going to hold on to it in the receptor on the surface of the cell. And then the T cells will then recognize that antigen presented in that MHC2 receptor on the surface of our antigen-presenting cells. And once that T cell recognizes and binds to that single antigen that it's able to recognize, it's going to be activated. That activated T cell can then trigger B cells to produce antibody, can also stimulate the production of cytokines and initiate the response from other T cells. Those cytokines can then attract other immune cells like our macrophages, monocytes, neutrophils to the location of the infection. And that is the end of section 13.3. Here are some checking in questions. Make sure you go through these and that you can answer them correctly. Also go back and review the learning outcomes. Make sure that you understand each one of those. Here's our critical thinking question. Victoria is blood type A negative, but her developing fetus is type A positive. The fetus possesses an additional protein marker present on its red blood cells that are not found on Victoria's cells. When fetal blood crosses the placenta and mixes with Victoria's blood, will Victoria's immune cells recognize these cells as foreign? Explain. So think about what we've discussed about antigens and substances that are good antigens or immunogens. Construct your answer, and then you can compare your answer to mine on the next slide. And hopefully this video was helpful.